What makes embryonal and yolk sac testicular cancers unique? Imagine discovering a lump in the testicle that turns out to be a type of cancer with its own special features. Embryonal carcinoma and yolk sac tumor are two distinct types of non-seminomatous germ cell tumors of the testis, each with unique biological and clinical traits that set them apart. Embryonal carcinoma is known for its primitive, undifferentiated cells that look like early embryonic tissue. These cells are aggressive and tend to grow quickly, often spreading early to other parts of the body. They are usually part of mixed germ cell tumors, meaning they can appear alongside other tumor types. Embryonal carcinomas produce two important tumor markers, alpha-fetoprotein and human chorionic gonadotropin, which doctors use to help diagnose and monitor the disease. Under the microscope, these tumors show a variety of patterns, including solid sheets, gland-like structures, or papillary formations, and they often have areas of bleeding and tissue death. Because they grow fast and can metastasize early, they requiring prompt and aggressive treatment. Yolk sac tumors, also called endodermal sinus tumors, resemble the tissue of the primitive yolk sac in an embryo. They are most common in young children under the age of 3 and tend to be more localized in this age group, which often leads to a better outlook. These tumors produce high levels of alpha-fetoprotein, making this marker especially useful for diagnosis and tracking treatment response. Unlike embryonal carcinoma, yolk sac tumors are usually confined to the testis in young children but can appear as part of mixed tumors in adults. The origin of yolk sac tumors is not fully understood, but some genetic changes, such as alterations in the RUNX3 gene and overexpression of GATA4, have been linked to their development. Both types belong to the group of non-seminomatous germ cell tumors, which tend to occur in teenagers and young men in their early 30s. These tumors grow and spread more quickly than seminomas, another type of testicular cancer. They are often found as part of mixed germ cell tumors, which contain different cell types, making diagnosis and treatment more complex. Management usually involves surgery to remove the affected testicle, followed by chemotherapy and sometimes radiation therapy, depending on how advanced the disease is. Monitoring tumor markers like alpha-fetoprotein and human chorionic gonadotropin helps guide treatment and assess how well the therapy is working. In summary, embryonal carcinoma is unique because of its primitive, aggressive cells that produce both alpha-fetoprotein and human chorionic gonadotropin, signaling its presence. Yolk sac tumors stand out for their resemblances to embryonic yolk sac tissue, their high alpha-fetoprotein levels, and their tendency to occur mainly in young children. Both requiring careful diagnosis through tumor markers and tissue analysis and are treated within the broader context of non-seminomatous testicular cancers. Always remember, if you notice any unusual changes or lumps, consult a healthcare professional for personalized advice and proper testing.